All right, so we'll continue with the central nervous system discussion. So, the blood supply to the brain is really important because the brain uses a lot of your blood, oxygen, and glucose, which is your sugar. So, in order to get those nutrients, you need to have a rich supply of blood vessels in the brain. So, these are just fun facts, but your brain is only 2% of your weight, but it's taking 20% of your oxygen and your sugar. So it's using a lot of energy. The problem with this is that your brain cells end up being susceptible to hypoxia, which is low oxygen in the blood. So in the brain, there's no stored oxygen. In the muscle, we have myoglobin. It stores oxygen. In the brain, there's no energy reserves. There's no glycogen or lipids hanging out. So the brain needs constant supply of blood that comes with oxygen and glucose. So even just 4 minutes can lead to permanent injury. This is why we do CPR, to continue getting blood to the brain. The muscles can live for hours without oxygen and glucose, but the brain can only live for 4 minutes without. So that is why we do CPR. So we'll go through kind of each of the functions of the brain and talk about a few different systems. A lot of this is just kind of memorization, so maybe make yourself some flashcards about each part of the brain and each function. So the hindbrain and the midbrain, kind of the back here, mostly do your life-sustaining things. So respiration, which is breathing, circulation, heart, and digestion. The cerebellum in the back here is really important for movement. In the hindbrain, there's a system called the reticular activating system. And this is what is regulating your sleep and your consciousness. If there's damage to this part of the brain, this will lead to coma. Also in the hindbrain, there are areas that control heart and vasomotor centers, and then there's also some pain control. I'm sure some of you are familiar with this, but an EEG is an electroencephalogram, and this is measuring the voltage changes in your brain. So basically it's measuring your brain activity. So you can see here there are a bunch of different types of waves. I don't care that you know the specifics, just kind of know generally that an EEG measures brain activity. So multiple different types kind of show if someone is conscious, sleeping, deep sleep, stress, etc. And sleep is temporary unconsciousness. Coma is no possible arousal. The reticular formation system regulates your state of alertness, and then a lot of you are probably familiar with the circadian rhythm. That's basically your biological clock that tells you when you should be awake and when you should be sleeping. There are a bunch of different stages of sleep. You go through cycles of non-REM sleep and REM sleep, and the REM sleep is what makes you really feel rested. So if you notice that stage gets bigger and bigger as the night goes on. So that's why if we cut off our sleep at like 2.5 hours, 3 hours, you barely had any REM sleep, so you'll wake up not feeling very rested. REM sleep's really important for brain development, learning, motivation, all of that good stuff. So sleep is actually an active process. So during sleep, there are a lot of things going on in the brain to kind of heal it and help you transform memories into long-term memories. For those of you cramming right now, you need sleep. The cerebellum is this back part of the brain here. It's really important for balance, eye movement, coordinating your voluntary movement. So if you want to play catch with someone, it will plan and coordinate all the actions that you need to do to like throw a football or something. It also plans and initiates the voluntary movement as well. Looking at the midbrain here, something important here on the midbrain, something called the substantia nigra, which is kind of a gray line on the midbrain. This is part of the basal ganglia and leads to Parkinson's, and we'll talk about this later. The tectum, for lack of a better term, are these two butt-looking structures here. So on top is the superior colliculus, and on bottom is inferior colliculus. The superior colliculus tracks moving objects like that football you're playing catch with, and controls ocular reflexes. So for example, if someone like claps in front of your face, your eyes are going to blink. That's from your superior colliculus. Inferior colliculus is your reflex to turn your head when there's a sound. So for example, if someone slams the door in your house right now, you would look that direction. Also controls auditory reflexes. So there are little muscles in your ears that help protect your eardrum from damage. And if there's loud sound, they'll flex to protect it. The thalamus basically takes sensory information and relays it and integrates it into the cerebral cortex. Underneath the thalamus is the hypothalamus. The hypothalamus has a lot of different functions. It maintains your emotional expression, maintains your body temperature, your thirst and your urine, how much you're eating, controls your pituitary hormones, and controls the circadian rhythm, how you're sleeping. The pineal gland is kind of sitting right near, this is like the superior and inferior colliculus right here. Pineal gland is this little bump right here. It secretes melatonin and serotonin. Melatonin is often known because it's a supplement people take when they're having trouble sleeping. So melatonin gets released during darkness to tell people to go to sleep. Serotonin is a neurotransmitter found kind of like, it's like called your like happy neurotransmitter. The basal ganglia are cells kind of scattered throughout the forebrain, and what they do is they take all the signals for movement and they kind of edit them. So, for example, your brain will say, hey, I want you to lift your right arm. But when it sends that signal at first, there's a lot of excess information. So the basal ganglia kind of edit the information so that your movement is more controlled. So what they do is they inhibit incorrect movement. So we call this the brake hypothesis. So this is a process called disinhibition. So it releases ongoing inhibition and inhibits ongoing inhibition. So your system wants to be on. You want to be sending muscle signals, but the basal ganglia just stop the inhibition. Damage to the basal ganglia can cause two really important diseases called Huntington's chorea and Parkinson's disease. Huntington's chorea results in kind of like a dance-like movement, and this is genetic because the basal ganglia cells get destroyed. There's a protein that gets produced that destroys the cells. I don't care that you know the specific name of the protein, but if you're curious, it's listed there. Big symptom, uncontrolled movements. Here's a picture. You can see this like gray line right here, which we know is a substantia nigra. Parkinson's disease is very similar to Huntington's. The substantia nigra has dopaminergic cells that get degraded, and that results in a resting tumor and really slow movement. If you know someone with Parkinson's, you're probably very patient because they move so slowly because it's so hard for them to have the correct movement. So the basal ganglia are supposed to inhibit wrong movements, but because they're gone, they are no longer inhibiting, so individuals end up with kind of a resting tremor. There's a video of a lady with a tremor. It does not work as far as I know, but you can try putting in this link and see what happens. Read through this question, go ahead and try and answer it, and I'll go through the answers with you in a second. Feel free to pause. So the difference between raising your keys to put a lock to the lock and actually putting the key into the lock is that raising your keys is movement and holding your key to put it into a lock is holding still. So the basal ganglia are more involved in preventing movement than causing them. So when she's trying to hold her hand still to put the key into the lock, she has a lot of trouble and a lot of tremoring because her basal ganglia are damaged. Damage to the cerebral cortex, like a stroke, often results in a loss of function. Why does damage to the basal ganglia result in more activity rather than less? Go ahead and pause, write down your answer, and then we'll go through the results. So when there's damage to the basal ganglia, we end up with extra movement because the basal ganglia are supposed to be inhibiting movement. If there's no more inhibition, we get extra movement. Here's another video you guys can check out, looking at someone with slow movement here. Okay, I'm going to stop here on this video and then we'll continue with functions of the brain on the next one.